prepared a little presentation. So I'm going to share my screen and go over that with you guys. If that's okay? Oh, I can't share my screen.
Oh, hold on. That's me. Sorry.
Sorry.
Hold on. Multiple. Try now.
Okay.
That work?
Oh, there we go.
Yay. Okay.
Alrighty. Can you all see this?
You will be able to now. Okay.
Yay.
Yay.
All right.
Good job.
Great. It's my first time sharing a screen.
I know we're learning here too. So.
All right.
What
Okay. So
are you guys seeing her screen or my screen?
Hers.
Okay, good.
the phytoplankton and yeah, cool. So I'm Ross-Lynne Gibb. I'm a PhD student in South Africa. I'm based at the South African Environmental Observation Network, and I study at Rhodes University. Can you guys see the cursor?
Yes.
Yes.
Oh, cool. Okay. All right, so I study phytoplankton in Algoa Bay, and I'll get into where that is now. A little bit about me. I'm doing my PhD, as I said, in South Africa. I'm from a city called Port Elizabeth, and I consider myself an honorary mermaid. I love to spend a lot of time in the ocean, and this has been my passion for as far back as I can remember. So to put everything into perspective, for those of you who don't know, South Africa is at the bottom of Africa, is a little highlighted country. We have 11 official languages, and we're a very culturally diverse and rich country, and it's a fact that we're very proud of. So my official PhD title is Phytoplankton as a Tool to Map Responses to Environmental Change. All right, so it's quite a mouthful, but we'll break it down. All right, so phytoplankton, sorry, gotta take that down. As the base of the food chain, changes in the environmental community have a knock-on effect on the rest of the ecosystem. So a shift from more palatable species to less palatable species are mirrored up the rest of the food chain, and everything depends on phytoplankton, because if you look at the little food chain over here on the side, we have our phytoplankton, which are eaten by zooplankton, fish, predators, everyone else, even us. We depend a lot on fish. So the thinking is that phytoplankton are good indicators to environmental change, because these organisms are very sensitive to change. So you change the temperature, the community changes. You change the nutrients, the community changes. Yeah. So basically, I want to know who's there, what are they doing, what happens when the community changes, and what causes these changes that we see. How we do this is we go out on a boat into Algoa Bay. I'll show you where that is now. We send down this piece of equipment. It's called a Rosette CTD. So it gets its name from the Niskin bottles. Those are those big, tall grey things over here. Those are arranged in a rosette fashion. In the middle of those bottles are probes. They measure conductivity, temperature, and depth. That's where the name CTD comes from. And they also measure a range of other environmental variables. It all depends on which probes you stick in the middle. And how this bottle works, how this thing works is you open up the bottles. So you see on the top here, there's a little cap and a little string, and you connect it to the string on the bottom, connect them together, and you open them up, and then you send it down with the crane into the water, and each bottle is set to trigger at a certain depth, and this is controlled by a computer. So you connect the computer beforehand, you load the data onto the CTD, you send it down, and the bottles close where they're supposed to. And that ensures that we collect water at a particular depth, and not the water above it and not the water below it, because you send a bucket down, the water's mixing, so you don't quite get the same result. Then I go back to the lab, I filter all the water so that I can extract the DNA from these samples. And I collect eDNA. So every organism has its own DNA barcode, and the method of taking those sequences and looking at them is called barcoding. Method barcoding is when you take an organism or a few organisms, selected organisms, and you sequence their genes all at once, whereas I do eDNA, which is environmental DNA, where you collect a sample of water or gut content from a rat or whatever the case may be, and you sequence all of it. And so what this does in the ocean is it tells us if a fish swam through, or it tells us if there was a whale, it tells us if a bird pooped, that kind of information. But there are issues associated with eDNA for doing fish identifications and stuff like that, but it has been a very useful tool in the study of phytoplankton. The only issues we have is zooplankton being organisms with lots of cells and phytoplankton being microscopic organisms with single cells, is that sometimes the zooplankton or the fish or the other organisms overshadow our phytoplankton. So we still need, this is some pictures of barcoding. We still need visual analysis. So we still need to make slides and dye the organisms and look at them under a microscope and spend the time counting them, so that we can be sure that these DNA barcoding methods are giving us the right information. And the hope is to one day move to a point where DNA barcoding is trusted, and these high throughput methods will save us a lot of time, because counting these cells is not an easy job. It takes a lot of time. So how I do this is I have these chambers, they're called Utamol chambers, and you put your known sample of seawater in that's been preserved. So the CTD is for my barcoding stuff. For this, I just collect a grab of water like you guys do with a net, phytoplankton net, and they get preserved in glutaraldehyde. And then I put my known sample in this tube, and I dilute it with some water, and I put in a dye. It's called rose bengal. It gives it this pinky color. And then what we get is this. So this is Noctiluca. I'll talk a bit about him just now. He's one of our harmful algal species, but he's a notoriously difficult species to spot, to see. As you can see all around the edges, they're quite thin, and it's like a see-through bubble. So they're really hard to see. And what the dye does is it sits along there and helps us to see him, and helps us to see everything that he's engulfed, because these cells engulf other things. Okay, now around South Africa we have two main currents. We have the warm Agulhas current down the east coast and the cold Benguela current on the west coast. And I went on the SA Agulhas 2 in 2018 and I sampled the Agulhas current. We wanted to know what species are in the current. So what we did is we collected samples on our way to the current. So you see the Benguela runs over here, and the Agulhas runs over here, and we collected these samples on the way there. The green spots, blue spots are in the current, and the little red spots are on the way back. So we chose these sites because they're important to Algoa Bay, which is my main study site over here. Now the reason the Agulhas current is important to the bay is because as you can see here, the Agulhas current intrudes into the bay. And what that does is it not only changes the chemical makeup of the bay, it changes the temperature, it pushes cold, nutrient-rich water from the bottom of the ocean up into the bay, and it also introduces biodiversity. So we wanted to know, how does the Agulhas current influence the Algoa Bay system, and can we use phytoplankton diversity to map the current intrusions into the bay? So is it different to the bay? And when it goes into the bay, does the bay look like the current? So Algoa Bay is down here, and relative, if you look at South Africa, it's over there in the little corner. And I have 3 sites within the bay. So I have the 2 extremes and an inshore site. And what we find is our bay is mostly dominated by things called diatoms. I'm not sure how familiar you are with them, but they're made of silica, and they're really pretty to look at. And these are all single cells, but if you can see in this little drawing, a lot of them like to form communities, and it's really cool. I think it's really cool. And I actually have a really cool video. I couldn't find it today to put it in the slideshow, but I have a really cool video of them working together to move around in my slide, which is really cool. Okay, and some of these species, this one down here is called Bacterial Strim. This one's Rhizosolenia. And this one's really cool because you see these different shaded areas. It's actually undulated. So this one is indented, whereas this one is raised, and then this one's indented. So that's like wavy. So if you look really carefully, you can actually see it. It's very cool. And then this one over here is called Thalassosira, Agnanthes, Coconeus. I'm not going to go through all of them because it would take us forever. But they're really cool and they're photosynthetic. They are said to absorb more carbon dioxide than the trees in the Amazon. They're also said to produce more oxygen than trees. And that's quite remarkable because there are these tiny microscopic things that nobody really knows about and nobody really cares about. Yet they're so important for our own survival and our own, the survival of our ecosystems as well. So we also have Dinoflagellates. So our two HAB species, we have Lingulodinium and Noctiluca. I'll get into what those are now. They fit in with the dinoflagellates. They are dinoflagellates. This one over here is called Protoperidinium. This is Porocentrum and Serratium. Those are our dominant species within the bay. So these are all photos that I've taken. It's quite remarkable to see these things under the microscope. And we get some things called silicoflagellates. I think these are very cool. And they form all these pretty shapes. But despite them being pretty, they're very, like I've said before, they're very important components of our marine ecosystem, or our aquatic ecosystems actually, whether it be marine or freshwater. And even though they're pretty to look at and they're these tiny little microscopic things, they play a vital role. And we thinking that with climate change and all these issues, environmental issues we're facing, that these will be critical indicators in the changes that are seen. Okay, so in 2014, we had our most extensive harmful algal bloom that I think we've ever seen. I stand to be corrected. So a harmful algal bloom, for those that don't know, it's when a particular phytoplankton species, so phytoplankton, a particular species is favored because environmental conditions are right for that particular species to grow and reproduce. And then it takes over the community, it outcompetes everything else, sucks up all the nutrients, and it causes quite a bit of trouble. So in 2014, we had our first bloom of Lingulodinium polyhedra, and like I said, it was extensive. I think it was something like 500 kilometers. I think that's about 300 miles or something. So that's quite huge. And relative to our coastline is not that big, relatively speaking. So that was quite a thing. And here's a nice aerial photograph of what it looked like. And the public quite enjoyed it because it's pretty and bioluminescent. And everyone was running around in the sea going, oh, it's so beautiful. And it is, it's very beautiful. But then there are the negative effects. So harmful algal blooms are known to have direct effects such as fish kills or human toxicity, some species. Other species have indirect effects where they change the phytoplankton community. So remember I spoke about the diatoms and the dinoflagellates. So the dinoflagellates, the harmful algal blooms are less palatable. Obviously, if things don't want to eat something that's toxic or going to harm them. The diatoms are more palatable. Most things depend on diatoms. And if the harmful algal blooms outcompete the diatoms, then the other species don't really have much to eat. And then, I don't know, they leave, the community changes, things like that. So they're a problem, and we need to figure this out. So this is a photograph I took in February 2018 from the back of our boat. So you can see The water's quite red. It's obviously not as bad as the 2014 example, but it's still there. And so this bloom has been occurring every year since 2014. And that's quite a thing. And the reason this happens is because Lingulodinium forms spores that settle into the sediment. And when conditions are favorable, they bloom again. So we kind of hit a point where we don't know if we'll ever get rid of them. We also don't know too much about them. So it's very important that we understand them. Then we have Noctiluca. Noctiluca's always occurred, so this is Noctiluca scintillans. There's a red species and a green one. This is the red one, I think. Yeah. Anyway, so Noctiluca is known to engulf other species, especially diatoms, because they don't have any means of photosynthesizing. So you know how plant cells photosynthesize, so do phytoplankton, but Noctiluca can't. So it engulfs bacteria, sorry. It engulfs diatoms that do that for it. And interestingly enough, if you look at this photo, those little round things aren't diatoms. Those are Lingulodinium spores. So we're unsure what function that serves, if any, and we're not sure what that does to the spores. We're not sure what Noctiluca gets from that. Yeah, so that's some figuring out we need to do too. But this has, it's a very interesting find, and perhaps we can use this information. So that leads me to the next thing. So by doing my PhD and seeing what phytoplankton are there and how the community changes and things like that, so we've definitely seen when there's harmful algal blooms, there's an increased amount of Dinoflagellates and decreased diatoms, because the HABs outcompete the diatoms, and then other things can thrive that aren't normally able to. And so this has kind of given me a keen interest in harmful algal blooms. So for my next thing, I'm going to be doing a postdoctoral fellowship where I study Lingulodinium polyhedra and Noctiluca scintillans. And the idea is we're going to culture them, and we're going to try and germinate Lingulodinium cysts. This way we can measure growth rate, we can manipulate the nutrients and the temperature and things like that to see what happens to them. And then by culturing them and growing them, we have samples to do sequencing with. The issue with these HABs occurring sporadically is that we're not always able to get the samples that we need to monitor them. So by creating the environment in the lab, we can then monitor them. We have enough samples to sequence. We can test for toxicity. There's been a lot of evidence that's shown that different strains of the species from different places have different levels of toxicity. I found papers that have said they're very toxic, and I found papers that said they only have a little bit of toxin in. So the toxin they produce is a yeastotoxin. It's been linked to some shellfish poisoning, but many people say the levels aren't high enough to be dangerous. But given the different toxicities between different strains, we're not sure what our ones do. We're not sure if our ones are toxic and how much toxin they have. We also do not know what natural products they may produce. Are there things that they produce that are useful? They found a lot of useful compounds in sponges in Algoa Bay. Very useful compounds. The issue with sponges is they don't have a very high turnover rate. So harvesting sponges for their chemicals is very unsustainable. But phytoplankton, if they can be cultured in a lab, then one could have a sustainable way of harvesting natural products. So the study of natural products in things like microalgae or phytoplankton has become increasingly important. And this helps us, there could be a potential for antibiotic resistant microbes or drugs to fight antibiotic resistant microbes. So who knows what we'll find, but that's kind of our mission at the moment, to understand these species and what they're doing in our bay, and can we use them? Can we culture them and use them for something better? Thank you, that's it. And then I thought after this we could have a discussion. You guys can ask questions and we can chat a bit about phytoplankton. Hi.
Hi. Woo! I'm going to unmute everybody. I'm unmuting everyone. If you want to mute yourself again, that's fine. But I'm just going to unmute everybody again. Yay! This was awesome.
Did you guys enjoy that? I try to keep it
Very
short and
much.
sweet so that it wasn't too much.
It was great. And okay, I have to do this. Okay, I'm sorry. I just can't help. All right, quiz time. Who saw any pictures of dinoflagellates and diatoms that they recognize? Judy, what did you see? What did you see in her pictures?
Dinophysis.
Dinophysis, very good. Who else saw anything they recognized from our samples here?
Astamphalis,
Yes, very good. Yeah, so we have
Navicula,
navicula.
Uh-huh.
Yes,
Oh, and our old favorite serratum furca.
yes, yes, yes. Did I ever tell you guys the serration changed its name and it's now furca? We don't call serration it serration anymore. I mean, we still do, and we will always. But in the scientific literature, the name got changed. So the name serration doesn't exist anymore. I don't know, just a little caveat here.
It's furca and just
Furca.
Fusus,
Oh, furca.
fusus and what about the other serrations? They're
They're
all
all
Lineatum,
yeah, they're not serration anymore. It's horrible.
holy moly.
We have another presentation just on taxonomy, but this was
Welcome
great.
to science.
Yes, welcome to science.
That will go on for hours.
Right. I have so many questions, but I'm gonna ask the volunteers and the citizen scientists and the other guests that we have here today to start with their questions. And if you guys have any comments too, please feel free to share.
Yeah, any comments would be great. I don't know how technical that was for you guys, and
Well, I would be really interested to know if this is affecting, these blooms are affecting the air quality onshore.
good question. These particular species are not known to do that, but there is a harmful algal bloom. I'm trying to remember its name now. I know the species name is Breve, B-R-E-V-E, but is it a serratum as well? I don't remember, but it's a particular harmful algal bloom that actually produces releases compounds into the air.
Yeah,
People actually struggle to breathe.
Carenia brevis.
Ah, there we go.
Yeah, from Florida. Yeah.
Yeah, oh yeah, they're having a horrible time.
And so I'm going to chime in because in Ross, we didn't have a chance to talk about what I'm researching and more details. That we didn't have that chance, but I'm studying the interaction between the phytoplankton and the atmosphere right above it. So I just happen to know that because it has to do with my research. So Judy, I don't know about those blooms in South Africa, but like Ross was saying, there are some blooms that do affect the air quality because phytoplankton are constantly exuding these components into the air, not just oxygen through photosynthesis, but they're exuding other components that will affect not only the composition of the atmosphere, but the pH of the atmosphere, the amount of particles suspended in the atmosphere, which then creates sea spray, and even clouds. Cloud formation is actually affected by the amount of phytoplankton in the ocean right underneath those clouds. Isn't that amazing?
That is amazing.
Phytoplankton will exude something called DMS, which is we can't see with our eyes, but that's being released by the phytoplankton, and that creates the nuclei for cloud formation. So it affects climate in a big scheme of things. And in terms of air quality for human health concerns, sometimes these blooms will be releasing things that can be, let's say, irritating to some folks. And Ross, we just had a big Lingulogenium bloom. One of my questions for you is, how long do your blooms usually last?
Oh, it depends. Sometimes it can be a whole month or two. Sometimes, so in 2018, it was like the whole of December and January, and then we saw it again in March, May, June, throughout
Oh,
the year. Depending
wow.
on the condition. And we're seeing it more and more now every year. They're getting longer, they're getting more frequent. Climate change is having a real fun time here with these guys.
Right. So here we are having a bloom. It's traveling. It's now dying off, but it lasted a long time, 2 months, more than 2 months now. And some people did report feeling uncomfortable because of the smells and probably because of some components. But we're not sure if that's being released by the Lingulodynium polyhedra or by the bacteria that are decomposing the bloom. So it's, like Ross was saying, there's so much stuff that we don't know about these blooms.
Yeah. It could also be things that are dying in the water.
Right.
Because with the cells decaying, what happens is they use up the oxygen in the water, and then fish and things like that die as well. Forgot to mention that just now. So apart from the whole changing the ecosystem thing and potentially toxic, they also could cause suffocation of fish. They sometimes clog fish gills because some, like Noctiluca is huge. It's like the size of a
it's big.
Yeah.
We get Noctiluca here, but not in blooms.
Just a little bit occasionally.
Yeah. And both our Lingulodynium polyhedra and our Noctiluca strains apparently do not produce toxins here in this area. So we do see hypoxia. With this bloom, there were some fish kills caused by hypoxia, but not because of toxins. And I understand that it's different for your region, which is really cool for us to be able to be talking to someone studying these organisms in a different location. So the genetics work that she's doing is crucial, right? Because she's looking at this genetic variability that can explain why some groups will be producing toxins in some places and why others aren't. Remember we talked about how these organisms form cysts, and those cysts deposit in a sediment, right? And so what any bloom that you have year after year will be, they will be in a way the same cells that have been stored in a sediment for a long time. It's different from, say, a place that is near a port, and say a new species comes in, or with those currents that Ross was talking about, that might bring new genetic groups, new populations into a specific area. But here we think that these blooms are always part of the same continuation of these cysts that get entrapped in the sediment. I have a question about the cultures. So you said you're going to grow. So everyone, I'm assuming none of you here tried to grow your own phytoplankton at home or in your lab.
Didn't go well.
Amber, did you try?
I tried when we were trying to get something so that we could, we do, Roz, we do field trips through the FLOW program. And we were trying to create like an at-home plankton kit that teachers could take back to their classrooms. And it wasn't very successful.
Right. It's really difficult. Some groups are much easier than others, but the two organisms that Ross is setting up to grow in the lab are really difficult to grow. Noctiluca and Lingulodynium polyhedra are known to be difficult to keep. So what's your plan? Are you going to isolate them from like a natural population, or do you already have isolates growing? Are you going to create a new media to grow them in? What's your plan?
Well, I'm going to have to isolate them myself. So the idea is in a hab, we're going to collect the water. We're going to take our filtrates for, our filters for our sequencing and stuff. Because obviously we have to monitor, the genetics, the population, all that kind of stuff as we go along, and we can compare it to the data I found in my PhD as well. That gives us a nice baseline. So I don't know, I've taken a bunch of literature and I've suggested this is going to be the temperature, this is going to be the water, this is going to be the medium. We'll see how it goes. Obviously I haven't tried it yet, but I'll keep you guys posted if you want.
We totally want updates. It's like Mission Impossible. We should do the dun 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 for her, because it will be a challenge, I know. And but we have faith in you.
Thanks.
Yay.
We can share our our strategies and our results and you guys find something that works and I'm struggling, I'll be happy for comments, and vice versa. If I get it right, I'll tell you guys what the trick was.
Right. So one technique is to actually have like a micropipette and a microscope, and then you draw, you look at the organisms under this chamber, the ultramol chamber that she was talking about, and then you try to kind of fish them out. Some people will have like a special micropipette that they use. Some people will just like put the little tube in their mouth and do the sucking underwater. Depending on the expert, they have different techniques. Some other people, they do the fractionation, right? So they'll take like a bloom and break it in smaller, dilute, and then grow them. Do you have like a plan of which one you're gonna try first, or not yet?
I'll probably try Noctiluca first because it's bigger, easier. It'll help me get used to the culturing techniques and stuff like that. And then I'll move on to Lingulidinium, which is obviously quite a lot smaller and probably a lot more finicky.
So I would think that getting the medium right would be really tricky. Getting the right soup, so to speak, would be really a trick to have just the right level of nutrients, and might differ for different species. I would think that would really be a challenge.
Yeah, but that's also what we're trying to find out. We're trying to find out what works. What is making them survive? What is making them do so well? What is triggering the germination of these cysts every time they bloom again? So the playing around with the nutrients and the temperature and the water, that's kind of the point. So we're very excited about it.
That's very smart, right? So this is it's a good example of a smart scientific design, because every time she fails, that data is good data. It's data that will contribute to her. So it's a good way to design your experiment, so that you know that even if that specific attempt fails, you're still collecting valuable data for what you're trying to answer.
Even no data is data.
Yes,
if you
even no
do a
data.
survey and you don't find what you're looking for, you can be like, well, it's not there. It's still data.
Very cool. Very cool. Any other questions?
Are harmful algal blooms increasing worldwide, or just our ability to detect them?
Oh, that's a good question. Not actually sure. Well, they are increasing in frequency that I've noticed. As long as I've lived here, I've noticed that they're increasing in frequency, and the duration of the blooms are longer. But before me, I can't really say. It might be a case of our technology is better, so we're just spotting them better. But also, I mean, a harmful algal bloom or red tide is quite easy to spot if it's inshore. So I think there's a lot of good observational data from public and citizen scientists like you. Just to say that they were there.
It could be a bit of both.
I think so.
Yeah, I think so. And I think now there's a consensus from this field that it is increasing in frequency and toxicity and extent, Jerry. So I think there's a few papers that came out a few years ago that I've seen, because that's a question that has been asked by scientists since I've started in this field 20 years ago. And so a group of researchers decided to look at the data existing, like Ross was saying, like we can answer that question from a personal perspective of what we as individuals have observed, but we can also go back into the literature and try to see what data is available. Again, what Ross said about records that exist, and some of these records exist for hundreds of years. Some places have been recording the occurrence of harmful algal blooms for a long, long time, even if they didn't have the capacity to identify the organism or to measure if they didn't have the technology or the knowledge. So with that kind of like study method to look at previous data, they've concluded that yes, that there is an increase in the number of harmful algal blooms happening around the world, number of places in which they're occurring, size, like area covered, toxicity levels, and duration. And I can forward that paper to all of you. It's a scientific paper. It's a little bit tricky to read, but we can definitely maybe have a discussion group on that. And some of the reasons have to do with climate change. not just the warming of the temperature, which stimulates phytoplankton in general, but also because of the stratification of the water. Remember we talked about how with the warmer ocean, you have the layers of ocean water, they become more stagnant. And so that creates a perfect condition for bloom to form because you have nutrients, warm water at the top, lots of sunlight. So good question, Jerry. One that we should vote on that. Maybe we could do like a follow-up just on that one topic.
That would be cool.
Yeah.
Another thing we hoping to answer is whether, so the different strains having different toxicities and things like that, is that environmentally driven or is it genetics? So that's also something we hope to answer. Comparing our environment to your environment and our toxicity to your toxicity could give us really important information on whether it's environmentally driven, these differences, or whether it's just genetically they're that way because they live over here, you know.
So I know nothing about genetics. Can you tell us again about I really enjoyed this slide that you showed the different types of genetics studies that you do. How complicated is it to run these, to do these scannings? And how did you decide which type of genetic technique you are gonna use?
So it's very difficult. So I don't come from a genetics background either. I never did it before my PhD. Kind of jumped into a PhD with stuff, a whole heap of stuff I've never done. I wasn't even a phytoplankton before this, phytoplankton person before this. So it is very tricky. The process is, so the reason we chose eDNA is because it's easier for phytoplankton. You don't have to go and isolate the cells and culture them, and so it's easier to give us an overview of who's there, because the issue with trying to culture them is you could miss some that didn't grow. There are species that cannot be cultured, so you're not even going to know that they're there. So we wanted to try this method, and we've been exploring different primers. So what a primer is we have a process called I can't even say it. It's called PCR for short. But what you do is you put in a primer. So a primer is a short piece of genetic material that basically telling it, I want this piece. Okay, so you're telling them, the machine that you want this piece. Put in your primer. You also put in a bunch of other chemicals and stuff, and what that does is it makes copies. Sorry, okay, there we go. So what it does is once you've put all the stuff in, you put it in a machine that, to explain it simply, the machine changes the temperature, and the temperature affects the process, and that, it's a whole cyclic thing. But long story short, it makes copies of these little pieces that are a copy of your primer. So it takes the primer and it says, okay, on this strand, we have it over here. Okay, cool, take it, make copies, So it makes copies of all these little pieces of DNA. And then that helps us down the line when we sequence it. The machine can then read those and then tell it, gives us back sequences. And then we go through a whole curation process where we take out the junk and all the stuff we don't want. We make sure all the sequences are aligned, because you cannot compare pieces that are not the same. Because then it's going to tell you it's two different things when it's not, I think. And so then you curate it, and then you can finally use the data. And then we also compare it to a database. And the issue with the database is not everything is in the database. So you get a lot of unknowns back. So the issues here are with the PCRs, you can spend an entire day doing PCRs and end up with nothing because it didn't work. And that's why it's so complicated. And then eventually you get to a point where you've got it and you submit it for sequencing, and then something went wrong there, and then you'll start over. So it's a lot of trial and error and starting over. There are rules of thumb. So if it didn't work, then you try increasing the DNA or decreasing the DNA. You try adding different mixture of chemicals, whatever the case may be. But then, like I've said, at the end of the day, when you've got your classifications, you get a lot of unknowns out of that, and then that makes it really frustrating because you've spent all this time and then you get, oh, I don't know what you are. Okay, we'll just stick you into the unknown pile. So yeah, it's quite tricky and complicated, and especially if you're starting out and you don't know what you're doing, it's even more of a headache. But we've gotten some really, really great data. I'd love to show you guys, but unfortunately I can't because I haven't submitted yet. But we've gotten some really, really great data and we're very excited about it. So that will come up.
Very cool. All right, I muted all of you to reduce the background noise, but it's not symbolic, okay? You can unmute yourselves and talk. And just go on that little corner on the, what side is that? Left side. And then find the little microphone and you can unmute yourself. Or if you raise your hand, I will unmute you from here. But any other questions for Ross? Because I have so many and I tend to dominate the conversation, as you all know me really well. I don't do it on purpose. It's just like ADHD people. So please, any questions? Tom, have a question?
Can you hear me?
Yes.
Okay, great. Hi, Roz. Thank you so much for
Hi.
your presentation today. I was just wondering, what was your hypothesis about these harmful algal blooms? What conditions, what are the magic conditions that take place that cause, for instance, the 2014 bloom? And since you're seeing it over and over again, have you noticed specific things that are happening, whether it's the temperature? Fascinated by, is climate change having an impact, like Joanna was saying? And again, and the only reason I think that anybody's going to care about any of this stuff, is there an impact on the local economy? Is fishing impacted, like you were saying, when there's fish kills? Is that impacting areas around? Is it decreasing food supply for people? Is it causing increases in prices for any commercial fish that they catch in the area beyond the scientific value?
Sure, good question. So in terms of the harmful algal bloom, first of all, the hypothesis is that increased temperatures and decreased wind speeds from the time led to this bloom in 2014. So that's the hypothesis. From my PhD, I only have two harmful algal bloom periods. That's just because of resource constraints, and you can't always go out when there is one. That's what's led to this whole culturing idea, is because they're opportunistic, and we have a very dynamic bay, and we don't always have the best conditions. So we have a lot of heavy winds and big swells, and our little boat can't handle those very well, and our equipment like the CTDs can't handle the conditions very well. So that's what's led to us wanting to culture them, because we've been struggling to grab them. And so that's where my postdoc comes in, is we're going to try and answer those questions as to what drives those changes. So I have a year's worth of data, but only 2 harmful algal blooms. It's quite frustrating. And then it happens, and you go out, and then it's gone. Missed it. And to answer your other question, our bay is very commercially important in terms of fishing. A lot of commercial fishing happens within and around our bay. We actually have 2 harbors within that tiny bay. So it is very important commercially. The impacts on fishing is not quite clear yet, because nobody's asked the question. Nobody's done the research yet. Nobody's collected the data. But people have said that they struggle to catch the same yields and as many fish when there have been blooms. And they're also not sure about the fish that they catch. They're reluctant to catch fish because it could be toxic. We don't know. So I'm hoping that my research going forward will contribute to all these questions and give us an idea of what's the way forward, and whether or not the fish collected at these times are safe to eat, whether or not the fish are dying because there's little oxygen, whether or not they're dying because their gills are getting clogged, or whether or not they just leave, because I'm not up for this. I'll go find my food elsewhere. We don't know yet.
Great question and great answer. Yeah, so interesting how it affects, the whole structure of the community and the human population too. So very cool. Any other questions?
I had a question. So Rosie, you mentioned that before you started your PhD research, you were into phytoplankton. What got you into looking into the genetics of the phytoplankton in South Africa?
So that was a gradual thing. I studied botany and zoology as an undergrad in my honors. So honors is our 4th year. I don't think you guys have that. I studied rhino as my project.
I'm sorry, a study of what? Say it again.
Rhinoceros.
Oh, rhinoceros. Wow. Now that's a huge change in field.
Yeah.
Okay. I just had to make sure I understood. Okay, you were saying rhinoceros. Okay.
Yes. The land animal. So the reason for that though was the mentor. I wanted a particular mentor to teach me how to science. And unfortunately, he was a terrestrial person, so terrestrial being on land animals. So I did it. I studied rhinoceros. And then the first opportunity I got, I went back to marine. So for my master's, I studied things called stromatolites. So for those of you who don't know what they are microbial mats. So bacteria like cyanobacteria take sand and bind it together, and they make these formations. And within these formations, there are communities. So in the mat itself, there are little invertebrates and microalgae that live in there. And then these formations also make these pools. They make little walls that contain water. And then there's fish and crabs and all sorts of stuff that live within these pools as well. So I studied the idea of microhabitats, because within a stromatolite system, you get different types that look different and their consistency is different. Like some of them are soft and buttery and condensed, whereas others look like ciabatta bread with all the holes in it, and it's like rock. You know, you struggle to take a sample.
Can I just take a little pause here to connect this with something that these volunteers know really well, because, you know, they not only work on the beach, they work on the wetland as well. And so what she's describing is something that you might have seen in our wetland, a different version, slightly different version, but there's microhabitats that form where you have detritus, where you have the algae growing. You guys all know what I'm talking about? When you look in the edges of the wetland? Yeah? So she's talking about, you know, that concept of microhabitats forming because of detritus and algae and bacteria that now serves as food that makes that habitat so rich. Sorry, I had to interrupt to connect with what we know, but yeah, go ahead.
No, it's good. It's good. So everyone's on the same page. Yeah, so I compared the microalgae and invertebrates between these microhabitats, but it was a lot of counting and microscopy work. And then I thought, you know, it would have been nice to sequence these. And then I met my current supervisor, and she does this kind of stuff. And she was looking to do work in Algoa Bay, and I was like, can I do a project with you? And then she was like, yes, let's do a project. And then we came up with this project together. And it's been really great, really, really great. So that's, you know, it's quite a long story. But what got me interested is that these tiny little organisms do so much. You know, they do so much, and we don't know much about them because nobody really cares. Everyone comes into this field like, I'm going to work with sharks, I'm going to work with dolphins. I'm guilty. I was the same. I was going to work with sharks. You know, that was my thing. And whole undergrad, I was like, I'm the shark girl. And then I've never even worked with them because the questions. So for me, it's more about the interesting questions and the finding out than it is about the animal itself. So I've always gone where the interesting questions were. I saw a presentation on stromatolites, and I was like, that's cool. This is what I want to do. And then I did the microalgae for the stromatolites, and I was like, hey, I really like these. These are really interesting. I want to do more on this. So it's kind of like been figuring it out as I go. But yeah, now I think I'm stuck. I think this is what I want to do now.
Very cool. Thanks for sharing. It's a great story. Very good story. Good question, Amber. Anybody else? I was gonna ask that a similar question on how you got to work. So thank you, Amber, for asking that, because we have some folks here who fell in love with phytoplankton after they retired from their regular careers. And as I told you, they've been with this, stuck with phytoplankton now. And then we have some other people who are starting their careers, who are considering what to do next, who are, you know, asking those questions right now, how to decide what they want to dedicate their adult lives and their brains and their, so this is great. I think that having the ability to talk about how we make those choices and how we move forward. Tom, do I see a question coming?
From me?
Yeah.
Oh, I'm thinking. So, bigger picture, you were talking about how everybody cares about dolphins and sharks. And I'm one of those people that loves dolphins and sharks and whales and everything that's massive and beautiful out there. And I love the micro world as well. But it's very difficult to get people to care and to be concerned about the micro world. Have you started that process? Are you working with groups that talk about the process? You started this whole conversation about how do we get the community involved, people of color, people that aren't all like us that love phytoplankton are really interested in it. That's a really tough question. How do we get more people involved in protecting the wetlands, in concern about the environment? You're a science person, not a science person. A lot of us are. But when you talk to the general public, it's different. And I was wondering if you had any ideas on that. How do we reach out to the public and grab their attention and say, this is so critical? Just the pursuit, like you were saying, the interesting questions, the pursuit of scientific knowledge. I think that's so important now than ever. Can you comment about that a little bit if you could?
Well, I think the most important thing is to talk about it, to let people know. I mean, a lot of people don't want to listen. It's okay, but we can talk to those who do. Every opportunity I get to talk to school kids, or last year I went to an elderly home, spoke to them. Hopefully they'll tell their grandkids about these phytoplankton things, you know. So every chance I get to talk to public, I grab it. We're encouraged in our institution to talk to public and school, especially school children, you know, try and get them interested in just science in general. So I think the answer is probably just to talk about it and keep talking about it and keep talking about it. And, you know, those that don't want to listen, it's their issue. But those that want to listen, I think they're the ones we need to go after and try and get everyone keen and interested. Unfortunately, we live in a world where life's tough, especially here in South Africa. We have a lot of poverty and things like that. So it's really hard to drill these issues, because if I can't feed my family, why must I care about you and your phytoplankton? You know, so it's really, that's what we struggle with here, is, you know, the poor people they want to just, you know, make ends meet. They want to put food on the table. These environmental issues that might affect their grandkids, you know, 4 generations from now, you know, I know it's affecting us negatively now, but, you know, they see it that way. They're like, well, I can't see it. I can't, you know, why must I care? And I really struggle with that. Because I understand if you're struggling to make ends meet, these issues aren't necessarily your priority. Your priority is, how do I get a job? How do I put food on the table? How do I, you know, so I think we need to be sympathetic to people's needs as well, and maybe try and figure out a way to make this matter to them. So, for example, harmful algal blooms affecting the fish stocks affects a lot of our poorer communities, because they rely on fishing for sustenance, or fishing for income, fishing for whatever else. And not just fishing, they rely on the ocean for a lot of other things as well. You know, there are people that harvest seaweed, there are people that you know, go out on ships and stuff, and these are important issues for them because it affects their livelihoods. And we have a lot of bunkering within our bay, and the odd oil spill, and so that's also a big issue that we try and tell people about. You know, we're having this impact on the environment, and it's going to impact our food. It's going to impact us today. It's going to impact us tomorrow. And I think that's maybe what we need to do. We need to relate our science to the guy on the street that's trying to make ends meet, the guy on the street who's unable to see the bigger picture, make it matter to him or her or them. I don't know if that answers your question, Sophie.
Tom? Yeah.
Yeah.
The next Yeah, I wanna again. So 2 bookmarks. One is Jerry's point about what's happening with harmful algal blooms. Are they increasing or not? We should definitely talk about maybe having a follow-up conversation about that. And then this one about how to make our science that seems so disconnected from the needs that people actually have, especially the people who are, like you said, struggling to just put food on a table and survive. Um, there's literature on that. There are a number of people thinking and studying that, and we should definitely be able to educate ourselves about what's already available, what techniques have been used in different places. I have not directly worked with this in a long time, but I had a brief experience in Brazil. When I was an undergrad, that's many centuries ago. Feels like a different life. But when I was an undergrad, I got to work for a few months as an intern in a turtle conservation program. And in Brazil, turtles were, when I was a kid, were still eaten. They were captured for human consumption, sea turtles. And so this group of scientists who used to just be straight up scientists, they decided to become conservationists. And they created these programs along the coast of Brazil. And the work was mostly not with turtles. It was to preserve turtles, but the work we did was with the community. It was with finding different job opportunities for these people to rely on. It was to help them to have better lives and to work to protect those same turtles that they were before hunting for their consumption. And we can say now the turtles in Brazil are doing better. The populations have come up. Turtle hunting is completely abolished. It doesn't happen anymore in Brazil. And you can visit those locations. The main source of income now is tourism and ecotourism specifically, to go visit these centers. So it takes effort and it takes years. We're talking about a program that took 20 years to actually start showing any results. It took public money, but it took private money. It took scientists, but it took also social workers, and so maybe we can talk more about that, of how. And then here at the Bolsa Chica, and Ross, I don't think we had a chance to talk more about this group here, but I'll send you a link to the video that tells a little bit about the history of this group. But this group has a legacy of reaching out to the community and advocacy. So I want to second everything that you said, and I want to invite everyone in this group to continue this discussion. This is an important discussion. I think it's timely and important. It's one that I'm having in the department here, where I am, about the role of scientists and everything that we're experiencing in the real world. It feels like science is not the real world. Science is like this little bubble, right? And we go in and we're just in our little science world. And I have to say, I'm very glad to see how that's changing here in my department. Just yesterday, I received a copy of an email that is being circulated by the students. It has been approved by most of my professors, and it's now going to go to vote by the department. And we are taking actions to increase our communication with our community. So scientists will now be committing hours of their week to be talking to community members, to be opening up their department to community members to come to us and to create that two-way communication that needs to happen. So we can learn from what people are doing and see how we can help.
Yeah, I think it's a very important issue, and we all need to, and I think you guys are doing a great job, citizen scientists, always a fan.
The thing that I find so fascinating is the fact that this is a global issue.
Mm-hmm.
It's not just local.
You mean the harmful algal blooms?
Everything. This whole thing, the scientific investigation, that it's a global thing that affects everybody, not just isolated places.
Right.
Yeah.
And I think what Ross was pointing out is that it affects everybody, but it affects everybody differently. And sometimes it's hard to understand that, especially if we are privileged to not worry about what we're going to eat tomorrow, right? It's hard for us sometimes to understand what the priorities are for someone who has to earn today, going to buy the food for their family tomorrow.
Mm-hmm.
So for us who are privileged with education and science and comfort and safety, it's hard sometimes to be able to connect with that other reality. We need to be able to find people who can help us translate, interpret, align needs and values, align what's possible and what's ideal. And that takes work. And a lot of times scientists are just too busy. They're trying to make tenure. It's not.
It's a really difficult question. How do you make your science important to the guy on the street? We know why it's important, but why is it important to, the single lady with a child who's trying to make ends meet? It's really hard, really, really hard question. Excuse me. And it's something that a lot of funding sources are asking for now. Why is your research important to the community? Why should we fund you over someone else? And I think it's a very important question, even though it's a very hard question. You'll need to think about it.
So how about we put some readings? So Ross, if you have any material about this topic that you want to share with us, please feel free to email. This week, I'll be collecting a number of articles that I've came across about this and examples of places that are trying to connect. And then we can all start thinking about what powers do we have as a group of people who are privileged to be able to meet once a week and talk about science and have some fun. What can we now do to help our community a little bit more? Maybe not more, but maybe in a different way. Maybe it's not about doing more. Maybe it's about doing a little different. Maybe what we've done so far was really good and it worked for what we needed then, but now what we need is a slightly different approach. You know what I mean? So maybe we can continue this conversation during the week by sharing articles. And if you all come across anything that you think pertains to this conversation and you want to share. Also, if this is something that you're not ready right now, you're doing other things, remember, this group is completely democratic and open. You can come in and come out whenever you want. It's, buffet style. Fill your plate with whatever you want, as much as you want. So I want to thank Ross. It's already 12:12. I don't know if, I think people, if you want to continue here and keep talking, that's fine. But I want to make sure everyone knows that we're definitely over our time. Thank you so much. It's late there, and
it's 9:15 at night. That's why it's dark.
There you go. So,
but thank you so much for having me. This was really cool.
Thank you for joining us. We really appreciate you taking your time.
And our meetings are usually at 7 p.m. So if you are suffering from insomnia any Thursday, join us, and we have a number of, I want to, just before people start leaving, I want to say that next week we're going to have a scientist who is going to be talking about ways to reach out the community through it's a small citizen science opportunity. We're asking people to mail plants, invasive grasses, to the laboratory because they're going to use those plants to measure change in air quality during the stay-at-home orders. So these scientists that are here in the department, I don't work directly with them, but they're my friends, and they are asking our help to get those plants. So if you have any friends anywhere else in California, outside of Orange County, and you needed an excuse to connect with them, this is the chance. Get the phone, call them in Fresno, Sacramento, San Francisco, anywhere in California outside of Orange County. We need them to mail invasive grasses. All they need is like one little piece of cheatgrass or one little piece of, wild oat. Roll them up, put it in an envelope. I made a video showing how to do that. And so next week, we're going to have the professor here to explain how they do that. They use isotopes and she's going to explain to us how is it that they can measure change in the amount of fossil fuel that is burning and the stuff that they find in the plant, in the body of the plant. Kind of cool, right? So tune in. It's 7 p.m. June 11th, next Thursday. And then the following Thursday, we'll have our very own, is that the following Thursday that we have Vic?
Must be.
Or do we skip one? I think we might skip one. But anyways, we'll be sending announcements again. So stay tuned. Next week, we'll go back to 7 p.m. And please come see how we can use plants, invasive grasses, to measure change in air quality. Talk about something that has to do with people on the ground, right? Like, we're all interested in knowing how air quality changes, and the scientists have a way to measure using plants. So, anyone wants to say anything before we end this meeting? No? Okay, so then,
Just thanks again for sharing the information. It's wonderful.
Yes.
Thanks for coming, guys.
Great meeting.
all right. So I'll see you all next Thursday, and let's keep the conversation going about how we as an organization can do better at serving and reaching communities who are normally underserved, underrepresented. How can we share the good things that we get to enjoy with them?
Can I ask, is there more information about that possible teacher training you talked about last week?
Yes, so we're working on dates. We don't have specific dates, but the board of directors of AMIGOS approved that summer institute last week. So we are gonna have the summer institute, woohoo! I'm thinking it's gonna be towards the end of July for two reasons. One, because we need to give teachers a little break. They're so overwhelmed right now, they can't be asked to be like doing work again. And then two, because we still don't know what's gonna happen in fall. We don't know if students are gonna be asked to be completely online or if they're gonna be in some kind of hybrid capacity. But the idea is that we're gonna be training 20 teachers, not training actually, we're gonna be helping them, giving them what they need to prepare their plan for fall so that they can adjust to the fact that they won't have access to field trips, in-person field trips. They won't have access to as many hands-on learning opportunities. I know Tom here. I don't know if you guys, has anyone visited Tom's classroom? Mr. Pfeiffer. When we talk about him as a teacher, it's Mr. Pfeiffer. Jerry, you've been to Mr. Pfeiffer's classroom, right? That place is like amazing. It's heaven. There's just so many things going on. He's got plants growing. He's got all these different kinds of models that the kids can play with. And I know he misses his classroom a lot right now. And I know that the students will be missing those opportunities. So this institute, Nancy, again, is to help teachers to find creative ways to adjust to the COVID-19 stay-at-home situation. We're going to teach teachers to do hands-on activities with things that the kids may have around the house, like, you know, experiments with things that they find in their kitchen, or experiments they can do in their backyard if they have a backyard, or if they can do like right outside their house somehow. We're going to be connecting teachers with National Geographic educators. So they're going to be sharing their resources with us. There's a new program called Earth School through UCI. So we're going to be connecting teachers with people who develop that program. And the idea is to serve teachers who are serving underserved communities. So that's kind of part of the selection process. Teachers who are teaching Title I schools will be given preference. It doesn't mean we're excluding anybody that we might be able to fold somebody in, but the preference will be given to applicants who don't have access to a lot of resources to begin with. Any questions that you have specifically besides dates and what I just said? So Tom and I are going to be working on a formal flyer, like a beautiful flyer, and we're going to be sending out to all of you and asking you to forward to whomever you think might be interested. It's gonna be for middle and high school teachers. Yeah, and we're super excited. And if anyone wants to help with that, we're taking volunteers to help with that. Okay.
Cool.
Thank you for bringing it up. Okay.
Right.
Anything else? No?
Oh, can I steal Roz's PowerPoint? Can we get your PowerPoint? That would be awesome.
Yeah, sure, no
Thank you.
problem.
Send
Just
it
ask
to
one,
Joanna.
just one favor. Please don't distribute any of the photos,
Okay, right, sure.
just because nothing's published yet, and I'll get in trouble because it's technically the institution's intellectual property.
Okay.
Okay, so just to make sure we understand, so you're giving us permission to see for our group, but we should not be posting online, and we should not be sharing it with third parties. Is that?
Yeah.
Yeah.
You can
That-
show other people in a talk that you're doing, but don't give it out there for everyone to have.
Okay.
Okay.
Because
So
then
he
somebody
can
else will be like, oh, this is my picture.
But Tom could show to his students.
I was just wondering, like in a marine biology setting
Yeah,
Okay.
you could.
or
Perfect.
environmental science. Thank you.
No problem. If you guys have any other follow-up questions, feel free to contact me. Amber and Joanna have my details. They have my Instagram and they have my email.
Okay, great.
Thank you so much. And if you guys have anything else, email us. If not, have a great weekend. Stay safe, stay sane. Take care of your heart. Take care of your bodies. Love you.
All right, thank you.
Amigos.